get started then. Welcome everybody to the CMCR Investigator Meeting, to which I'm pleased to welcome my colleague, Dr. Charles Taylor. Just a brief background regarding Dr. Taylor. He is Associate Professor of Psychiatry at UC San Diego and Director of the Positive Emotion and Anxiety Research Laboratory, acronym PEARL, right? He received his bachelor's degree in psychology at McMaster University in Hamilton, Ontario, and his PhD in clinical psychology from the University of British Columbia. His research over the past 20 years combines experimental psychopathology and clinical trials approaches in anxiety and depressive disorders to identify target processes that give rise to negative emotional states or inhibit the experience of positive emotional states, with a focus on developing and optimizing intervention approaches designed to enhance social connections and well-being. Dr. Taylor's presentation today, as you can see, is dose-dependent effects of cannabidiol in social anxiety disorder, a randomized experimental therapeutics trial. I might say it's special for me, Charlie, to particularly welcome you as a fellow alum of the University of British Columbia, a wonderful place, as I also graduated from there from med school. However, I am chagrined to say that despite our common roots and my efforts to leverage our alum connection to ask Charlie to whisper in my ear what the results of the study are, he has declined. So here we are. Welcome, Charlie, and we're all ears.
Awesome. Thank you very much, Igor. I'm glad you didn't peek at the slides. Can I please? Oh, great. So let's see here. I'll share my slide deck. All right. Can I get a thumbs up if everyone can see that okay? Perfect. Thank you so much. So I'm really excited to present findings from this study today. This is has been a very long journey into the cannabinoid space for myself and my colleague Murray Steen and our team of many of whom are on this call. And to my knowledge, this is going to be the first, at least that I'm aware of, dose-dependent test of cannabidiol in social anxiety disorder or any of the anxiety disorders. So with that, I'll jump right in. Acknowledgements, again, this is a collaborative project. My MPI, Murray Steen. A ton of people were involved in this project. I'm not going to go through and name everyone, but I'm really grateful. I mean, both for the formal and informal support I've received. This was a beast of a trial, and yeah, I'm really grateful for everybody's help, and I'm excited to share the findings. A couple of disclosures. These are some companies I've consulted with. I should let you know that none of this is in the cannabinoid space. I should also disclose that by way of background, I am not a cannabinoid researcher. I'm an experimental psychopathologist and a clinical trialist who's interested in validating and developing new and better treatment options for anxiety and depressive disorders. And this is the first time that I've presented this data outside of our lab. So I'm really excited to get people's input on what we found. So with that, I'll jump right in. So the psychiatric condition of interest and the focus of this research is social anxiety disorder. And this is one of our most common psychiatric conditions. It's characterized at the core is a fear of negative evaluation. This can come across in different social or performance situations, having a one-on-one conversation with somebody that you just met, or maybe an acquaintance or close friend, small group in situations, or performance-based situations where people might be observing you in some way. And again, the core fear is that the individual will act in a way, they'll say or do something, or show signs of anxiety that may elicit embarrassment or humiliation or some sort of negative evaluation. And as a result, the individual with social anxiety disorder goes to great lengths to avoid social situations, or to endure those situations with great distress and to try to mitigate their fears through a number of more subtle avoidance behaviors in those social situations. And not surprisingly, given the ubiquity of social interactions in our day-to-day life and the importance of social connections in our mental health and well-being, social anxiety disorder is associated with significant impairment across a number of different life domains in terms of work, school, social leisure activities, and overall health and well-being. The good news is that we have some relatively effective treatment approaches for social anxiety disorder. So our first-line evidence-based treatment options, we have cognitive behavioral therapy of different flavors, exposure to fear-provoking situations with or without cognitive restructuring is quite effective, and a number of different serotonin or norepinephrine reuptake inhibitors that are FDA approved for the treatment of social anxiety disorder. So that's the good news. The challenge, of course, is that response rates, if you depend on how you look at it, the glass is half full or half empty. Only about half of folks with social anxiety disorder display an adequate treatment response or recovery, especially for our medication options. There can be a non-trivial side effect profile, and this leads to a lot of people not being compliant with their medications or just not being interested in pursuing medication. And then accessibility is a challenge too, especially finding providers, both physicians and psychologists that can deliver evidence-based treatments to fidelity. So those are the challenges. And my colleague Murray Steen and I, how do we enter into the cannabinoid space? And really, it's the, I guess, fault of the CMCR. There was a call put out, I think it was in 2019, for pilot funding to study cannabinoids. And one of the areas of interest was anxiety disorders. And so we discussed and we decided to enter in that literature to kind of understand the landscape of what was going on. The first thing that really struck us is that, how common self-management of anxiety is with cannabinoid-based products. So what I'm showing you here, these are results of a survey in over 2,000 people who've been given authorization to use cannabis for medicinal purposes. And what the researchers did is they surveyed these folks in terms of what was the authorization for, and then they measured a number of different psychiatric conditions to understand probable diagnoses. And for our purposes, what struck us is that anxiety symptom management was a very common reason why people were prescribed cannabis. And you can see that almost half, right, 42% of people were given a diagnosis of probable social anxiety disorder based on a self-report interview. And this was really striking to us, is that nearly half of the sample reported that they had replaced a physician-prescribed psychiatric medication with cannabis. And so that suggests that cannabis self-management is exceptionally common for anxiety-related issues in the general public. The problem is when you look at the evidence base, there's not a really compelling evidence base to inform what type of cannabis-based product people should be using, what dose should they be using. So really people are making their own decisions without really strong evidence about efficacy or safety or tolerability. So that encouraged Murray and I to pursue this area. And we decided to focus on cannabidiol for anxiety. And the reason was severalfold. First of all, the literature, when we surveyed it, most of the work in the anxiety space had been looking at CBD for the treatment of anxiety. This is in part because THC contains more of the psychoactive component of the cannabis plant. At higher doses can be anxiogenic, so it can actually induce anxiety in people. That can be offset sometimes if you add in CBD with that. But most of the literature really focused on CBD, which is the non-intoxicating component of, or a non-intoxicating component of the cannabis plant. The other main reason that we decided to pursue cannabidiol is Murray said he had a guy that could get us CBD. And in fact, I think it was his neighbor. So that seemed promising. And so we decided to pursue that. So what does the evidence suggest so far about the anxiolytic effects of CBD? So this was a scoping review. I'll just sort of cut to the chase here. This was a scoping review published a few years ago. And the conclusion, and this was across all psychiatric conditions, including anxiety disorders, was that there's not enough high quality evidence to support the clinical use of CBD for any psychiatric condition. Okay. More recently, a systematic review and meta-analysis was published last year. A little bit more of an optimistic soundbite here. They found, or the authors concluded that there was substantial, significant impact of CBD on anxiety with a considerable effect size. So very exciting. When you drill down a little bit more, that conclusion is based on what I would argue is a relatively modest amount of literature. I'd say very encouraging. But when you look at it, only 2 randomized controlled trials have been conducted in anxiety disorders with a total of 49 participants. One of those trials was an acute dosing study, and one was a chronic dosing study over 1 month. So, when we looked at this literature, we thought, hey, there's some more research needed. So what I'm going to do now, I want to drill down into 2 of the guiding studies that informed our work. And this is the infamous, I'll put the adjective infamous Bergamaschi study. This was published over a decade ago. It's been cited over 1,000 times. And this was an acute dosing study where the investigators randomly assigned 24 patients diagnosed with social anxiety disorder, either 600 milligrams of CBD or matching placebo, and they exposed them to a public speaking challenge. And the public speaking challenge essentially is a way to induce that core fear of negative evaluation. It's a reliable, provoke stress, frankly, in most people, but definitely for this target condition of social anxiety disorder. And we know that when you put people with social anxiety disorder into this particular public speaking challenge, you let them know that they're about to give a speech in front of a video camera. You rate how anxious they feel. You then put them into that speech environment. You rate how anxious they feel. We know this reliably induces anxiety. And the key finding here is that when patients were administered CBD, that anxiety response was less intense. And this was both during the anticipation phase, so when they were thinking about the speech they were about to give, and in the middle of the speech as well. And you can see that bottom line there. Those are people without a history of anxiety or any psychiatric condition. And these folks were pretty cool, calm, and collected throughout the procedures. What was also striking about this study is the investigators measured the negative cognitions or self-talk that patients with social anxiety disorder often have. So these are statements like, I'm a loser, I'm going to bomb this presentation, the audience is going to think this is terrible, this is a demonstration of my incompetence. And these are the sorts of self-talk that maintains anxiety. And what the investigators found is that in social anxiety disorder patients who had received CBD, it essentially had quieted that negative self-talk. And you can see that actually people assigned to the CBD condition almost look no different from the healthy control group. So this was really exciting data. Obviously, small sample size. There were only 12 patients per treatment arm, but it was, I think, very exciting and has been obviously talked about quite a bit. So what about chronic dosing studies? So there's one chronic dosing study that I'm aware of in social anxiety disorder. This was in 37 late teens. They were 18, 19 years old with a diagnosis of social anxiety disorder. And they were randomly assigned either 300 milligrams per day of CBD or matching placebo. And the primary outcome was the Liebowitz Social Anxiety Scale. This is the gold standard clinical symptom measure for social anxiety used in clinical trials. And what the investigators found is that people assigned to the CBD condition experienced a significant and approaching large size reduction in social anxiety symptoms that was greater than placebo. So again, really encouraging findings. That green dotted line there at the 50 mark, that represents sort of a cutoff where we might say that people above that line have moderate or greater social anxiety symptoms. Sometimes in the field, we'll use that if people drop below that, we might say that they're in a responder, at least partial responder status. So you can see, at least at the group level, that there's still some room for improvement. maybe 1 month is a pretty short duration for the treatment of social anxiety disorder, which is a pretty chronic condition. But again, a question about dose here. We've got 300 milligrams, a question about whether that was in the optimal range. So that work then inspired Murray and I to submit a proposal to the CMCR seed funding. They rejected us, although the reviews were very positive. So, we didn't stop there, and we submitted a second application the subsequent year, and they rejected us again. Also very positive reviews. And then Igor reached out and he had a meeting with us, and he said, "Hey guys, I think this is really promising work." And he had some of the other CMCR folks there. We really think that you should find another opportunity to get this work funded. So we started looking, and what we found is that the NCCIH, so the National Center for Complementary and Integrative Health, had a program announcement focused on clinical trials for natural products that could be used as a potential intervention for some sort of health-related condition. And they were really interested in natural products that people are typically using in the community to self-manage symptoms. So we thought, hey, actually, this is a great fit. I showed you data that a lot of people are already using cannabis-based products to self-manage anxiety. We thought this would be a good fit. So let me kind of talk to you a little bit about the framework of both this funding mechanism, but also the sort of clinical trials development space that's happening in the field and across several of the NIH institutes. And this is, people use the term experimental therapeutics, experimental medicine. And the basic idea is that we have an intervention, and that intervention, we want to see it improve some sort of clinical or functional outcome of interest, right? So if you think about most clinical trials, we're looking at things like, did people's social anxiety symptoms improve? Did the level of disability or interference improve? That's sort of the end state that we're interested in. But the treatment is not directly necessarily targeting those symptoms. It's targeting some sort of disease process or mechanism that we think maintains those symptoms, that's responsible for fueling those symptoms, so that if we can modify that particular mechanism or disease process, that will then be responsible for clinical improvement. And so this idea about there's a treatment target, something that this intervention is going to modulate, and by modulating that, then that will improve the clinical end state of that condition. And the framework is it's a phased two-part funding mechanism. And in the first phase, what they refer to as the R61 phase, the focus is on dose-dependent target engagement. And so here, we're not worried about clinical symptoms right now. What we're worried about is identifying a treatment target that we think will be engaged by the intervention under consideration, and that there's at least some hint or evidence that that treatment target is responsible for the clinical symptoms of interest. If you demonstrate that in the R61 phase, then you move on to the R33 phase, where what you do is you replicate target engagement, but you do this by taking the dose that showed optimal engagement in the R61 phase, and you replicate that, and then you examine, does the degree to which that treatment target changed, does that relate to improvements in clinical symptoms? And if the answer is yes, that sets the stage then for a much larger scale confirmatory efficacy trial. So the idea here is, rather than jumping to a large scale trial where we're looking at symptoms, the idea is we're focusing on the disease mechanism, and then establishing that, yes, that disease mechanism shifts, and by shifting that, it is also responsible for shifting symptoms. So for our proposal, we focused on threat reactivity as our treatment target. And by threat reactivity, I mean, essentially, it's the constellation of cognitive, behavioral, and subjective emotional experiences that are expressed when people with social anxiety disorder are confronted with their main fear-provoking trigger. Okay. And so for that, I'll turn back to the Bergamaschi findings, right? So these researchers put people in a fear-provoking situation. They expose the patient population to the core fear of evaluation. They measured the anxiety response. They measured the cognitive response under that condition and under different intervention conditions. There have also been other trials, I should note recently, other companies that have been using this approach in the same paradigm, this public speaking paradigm in phase 2 trials and now phase 3 trials to look at essentially anxiolytic effects of a particular compound under investigation. So these are published findings from a phase 2 trial of Neuphoria Therapeutics showing separation of both doses of their compound under investigation from placebo during the anticipation and performance phase of this public speaking challenge. And this is a phase 2 trial from Vistagen examining anxiolytic effects of their intranasal compound that they also showed separated from placebo during anticipation and performance. And so that's really what we wanted to pursue as our main treatment target. There was one additional element of this funding mechanism that we had to focus on. The funding mechanism at the time also required a biological target. And I'll tell you a funny story about that a little bit later. But for the purposes of the funding mechanism, that we needed to also demonstrate that in this case, CBD, the treatment under investigation, engaged some sort of biological target or mechanism that we thought was responsible for maintaining social anxiety symptoms. And therefore, by engaging that target, it would be responsible for clinical benefit. So we turned to the literature. What target should we pick? Well, for those of you who hang out in the cannabinoid space, this is my read of the literature, that there are numerous and complex biological effects of CBD that could account for anxiety reduction. What we decided to focus on was the endogenous cannabinoid anandamide. And we did this for several reasons. So first of all, there was some, I'd say relatively nascent, but encouraging preclinical data and experimental psychopathology data in healthy humans showing that anandamide has an anxiolytic effect when you expose animals or humans to stress. So that was sort of the one piece of evidence. There was also a parallel line of research focused on FAAH inhibitors. Now, FAAH is an enzyme that breaks down anandamide. And so FAAH inhibitor, several companies have been pursuing these synthetic compounds that essentially prevent the breakdown of anandamide by inhibiting FAAH. And these compounds have been shown to reliably increase anandamide and to reduce stress reactivity. So some of this work was coming from Leah Mayo and Marcus Heilig's group. And then there was also a study, 1 month chronic dosing, daily dosing trial in patients diagnosed with schizophrenia at 800 milligrams of CBD, showing an increase in serum anandamide over the course of treatment. So what about anandamide and social anxiety disorder? So my colleague Murray Steen had been working with Janssen Pharmaceuticals to pursue the study of their FAAH inhibitor compound. And what I'm showing you here is results from their randomized controlled trial. And I want to highlight two things. On the left panel, this is showing essentially a very robust correlation between trough plasma concentrations of the FAAH inhibitor and increases in anandamide. Again, just showing and really driving home that these FAAH inhibitors reliably increase anandamide levels. And on the right-hand panel, this is showing clinician-rated improvement in people administered to the FAAH, assigned to the FAAH inhibitor group versus those in placebo. And what you see here is by week 12, almost a doubling of improvement or proportion of participants that were identified as improved who were assigned to the FAAH inhibitor relative to placebo. So this data again suggested that there are some links here with anandamide and social anxiety disorder that maybe we might want to pursue. And so for this trial, we then really had two treatment targets, the threat reactivity treatment target, and then the biological target, anandamide, that was a requirement of this funding mechanism. So the study aims were then to test the hypothesis that CBD decreases threat reactivity and increases plasma anandamide levels compared to placebo. And the second aim was to determine which dose of CBD produces a greater effect. We decided a dose range of 300 or 900 milligrams. The literature, again, not a lot of studies out there, anything 200 milligrams and less suggested to not really have much of an anxiolytic effect in the few studies that had tested it. Pretty consistent findings in the 300 to 600 milligram range. Above 600 milligrams, the evidence was a little bit more murky. Again, not a lot of people, or I wasn't aware of any studies specifically in anxiety disorders that had looked at a dose higher than 600 milligrams. There was some data in healthy controls exposed to a public speaking challenge that suggested that maybe higher doses, weren't as efficacious. But we had consulted with, at the time, Greenwich Biosciences, consulted the literature in some of the other psychiatric conditions, and thought that 900 milligrams might be the sort of upper range of the effective dose, and that's what we pursued. The study design, it's a phase 2, is a 3-arm trial. And for this R61 target engagement phase, we focused on a subacute 4-day dosing trial. And the rationale is we wanted to get people to steady state levels of CBD, and then to measure threat reactivity responses once people got to steady state. And the idea being that if we demonstrated that, that would then inform a chronic dosing trial. The idea being is that, the simulated anxiety provocation in the lab would then sort of be a model for what CBD might be doing to folks out in the real world when they expose different social triggers. We randomly assigned 61 patients diagnosed with social anxiety disorder, 57 of whom were dispensed the study drug, and all 57 of those folks completed the post-test. And so 2 things to note here. One is huge shout out to my study team. These folks are wonderful for really sort of managing the trial, taking care of our participants, making them feel like their contribution is valued to get them to that post-test. And 2 is, this is one of the benefits of doing a shorter term, more target engagement based trial where you don't have to worry about retaining people over long periods of time and attrition and things like that. Our randomization was double blinded, equal proportions across the 2 CBD arms and placebo. And we stratified randomization by sex because there's some data to suggest that there may be sex differences in response to cannabinoids. The study drug, we're grateful to Jazz Pharmaceuticals. We sort of ran into a bit of an acquisition challenge along the way. This was my first experience with something like that. Greenwich Biosciences had agreed to support us, and they were acquired by Jazz. And folks at the CMCR were very supportive of us during the year-plus long adventure to finalize contracting and acquire the product. But we did acquire the product. They provided both Epidiolex, which I should say is a plant-derived, 100% plant-derived, highly purified oral solution of CBD. That's FDA approved, and I think maybe still this only FDA approved cannabinoid product. It's FDA approved for treating seizures in young kids with some rare conditions. And they also provided matching placebo, which looked, tasted, smelled identical. And the dosing schedule was twice daily dosing for 4 days. We encourage people to take the dose during a meal. And this was based on earlier pharmacokinetic and PD data in healthy individuals to kind of maximize the dosing regimen. Adherence was exceptionally high. I think out of a possible 399 doses that people were supposed to take across the trial, I think there were 3 total missed based on self-report. And one important element of the trial, and this was a bit of a challenge, but we figured it out, and I think I have to thank Murray and his wife for working their way through this one. The dosing needed to be blinded, right? We don't want people to know, okay, I'm on 300, I'm on 900. We're trying to mitigate expectancy effects. And of course, the challenge is that people are self-administering. They're drawing out the quantity of CBD or placebo, whatever the liquid is, the bottle that they've been assigned to. So you would know if you're pulling out more liquid, you'd sort of know, okay, great, I'm assigned to the higher dose. So the way we blinded this is we gave people 2 bottles. And one of the bottles, if you were assigned to a CBD condition, contained CBD, and the other bottle contained placebo. And what we did is we labeled the bottles 1 and 2, and the instructions for how much solution to draw out of each bottle depended on the dose that people were given. So all people knew is like, all right, I took, a milliliter and a half out of this bottle and 4.5 out of this bottle. They don't know active placebo, or in the case of the placebo condition, it was placebo. And that allowed us to maintain that dosing blind and again, mitigate expectancy effects. The sample, pretty typical. We were trying to sample the range of adults 18 through 70, tended to be a little bit younger. Not surprisingly, there's fair interest in the cannabinoid space with the younger population. 2/3 identified as women. We had pretty decent racial and ethnic diversity, which is consistent with the San Diego area. In terms of severity of social anxiety symptoms, the score represents people on average in the moderate to severe range of social anxiety disorder. So these folks are pretty impaired by their condition, and depression was in the mild to moderate range. All right, so let's drill into what we found. So what I'm going to show you here, the main comparisons, or sorry, let me tell you a little bit more about the outcomes first, and then I'll drill into what we found. So how do we measure threat reactivity, right? So I told you a little bit about the public speaking challenge. Let me tell you exactly what this looked like if you were a participant in our trial. So you come into our research clinic, you acclimate to the environment, you hang out for a little bit, you fill out some baseline assessments, we collect blood plasma, which we use for our biological target. And then the experimenter comes in and lets the participant know that in the next part of today's study, they will be delivering a 5-minute speech assessment. And we let the participant know that they will be given 5 speech topics to choose from. They can choose one of the 5 topics, and they'll have 2 minutes to prepare their speech, during which time they can jot down notes. And after the 2 minutes, they'll be taken into another room, and they'll be asked to speak for 5 minutes on that speech. They won't be able to use notes during that 5 minutes. There'll be a small audience of 3 people who will be observing the speech. And then the speech will be video recorded, and the performance of the speech will later be rated for its quality. So you can imagine if your core fear is negative evaluation, this particular standardized context does a really nice job of kind of priming all of those main triggers. Our primary outcome was the subjective units of distress scale, or SUDS for short. And so this is a fairly intuitive scale, ranging from 0 to 100. People are asked to report at the current moment, how much anxiety or distress are you feeling? We give people anchors so they can have a sense of kind of what we're looking for in terms of that. And we're measuring this throughout the procedures. So what I'm showing you here is essentially when we're capturing these SUDS ratings. So we're capturing it at rest, we're getting it throughout the anticipation phase at 1-minute intervals, and then 1-minute intervals throughout the public speaking challenge. We also measured state anxiety inventory with another measure, and then we measured that negative self-talk, as was done in the Bergamaschi study. So our primary outcome, we look both at the anticipation and performance phase. And the reason is that both are relevant to understanding social anxiety pathophysiology. Anticipatory anxiety keeps people from going into social situations. Performance anxiety kind of disrupts performance, makes people want to escape and not go back in it. And because of the small sample size, we pre-specified an effect size difference, a standardized mean difference of 3 or 0.3 or greater in favor of either CBD dose versus placebo. So what I'm going to show you now in the results section, I'm going to kind of walk through these. Whenever you see, you'll see graphs of change from baseline in placebo versus CBD in each of the CBD arms. And then you'll see an effect size chart, and you'll see where we met this effect size benchmark is highlighted with a red square. All right. So this is the 300 milligram CBD arm, and this is both anticipation and performance phase. And what you can see here is that in both cases, you see a larger reduction in anxiety during both anticipation and performance, but it was only meeting our effect size benchmark in the performance phase. So encouraging, technically, this is a go decision here for the 300 milligram dose. What about the 900 milligram dose? Now we see across both the anticipation and performance phase, we see a medium-sized, larger reduction in anxiety response from baseline to day 4. Okay, also very encouraging. What about across our other measures? Now things get a little more interesting. So this is the Spielberger State Anxiety Inventory measured in anticipation and performance. And yes, those are two lines drawn there. The 300 milligram group did not separate from placebo at all, whereas the 900 milligram group did consistently. And you can see there during the anticipation phase, that was a large size reduction difference in anxiety during the anticipation phase. And what about those negative self-statements, right, that Bergamaschi found in their study? Again, a trend, right, toward greater improvement in the 300 milligram group. It did not meet the effect size benchmark, but in the 900 milligram group, it did. And again, it's approaching a large-sized reduction in that negative self-talk that often characterizes people with social anxiety disorder. So those are our core primary and secondary outcomes, right? Really sort of showing us that, okay, on the primary outcome of SUDS, we see a hit across both anticipation and performance phase, maybe a little more consistent in the 900 milligram. Once you get to the secondary outcomes, you see that the 900 milligram dose is more robust in response. And what about the exploratory outcomes? So we measured avoidance behaviors. We measured the probability and cost of people's, idiographic fear-related outcomes. We measured positive self-statements. All I want you to take away from this graph is the green highlighted here, which again shows where CBD differed from placebo. And the 900 milligram dose was almost a clean sweep here. There were only two outcomes where it did not show superiority versus placebo, where again, you see a much more modest or less consistent effect in the 300 milligram dose. So what about the dose comparison? So right now we've established, okay, relative to placebo, we see efficacy meeting those effect size benchmarks we set. We just said, okay, we're going to pursue the more efficacious dose if there's an effect size difference of at least 0.2 or greater between the doses. We thought there'd be at least some efficacy in the lower dose, but we need to show at least a 0.2 or higher difference. Assuming safety profile, side effects look similar, which I'll get to momentarily. And again, this is a lot of data. All I want you to take away from this is the green, right? That pretty much across all of our outcomes, we see that the 900 milligram dose outperformed, showed a greater reduction in threat reactivity relative to the 300 milligram dose. So for us, this was really kind of driving home at the between group level that there's a dose response relationship. Okay. We also collected blood plasma and we measured both CBD, plasma levels of CBD, and 7-hydroxy CBD, which is one of the major active metabolites of CBD. And we correlated doses of those, or sort of levels of those on day 4, with change in anticipatory and performance-related anxiety. And what we found is that larger plasma levels of CBD and 7-hydroxy CBD correlated with larger reductions in anticipatory and performance anxiety. So we see both at the treatment arm group level and we see at the correlational level a dose-response relationship. So we're very excited. We're ready to party, but we forgot about anandamide. All right, so our hypothesis here, simple. We've got one outcome, test the hypothesis that CBD increases plasma anandamide. And I'm emphasizing increases here because that's what the literature led us to believe we would see. We collected resting plasma anandamide 10 minutes after people had acclimated to the lab setting. We had our standardized mean difference benchmark. We actually had a higher benchmark here because we thought that maybe this endogenous cannabinoid would be more proximal to the intervention than threat reactivity. So what did we find? Well, we did see separation of CBD from placebo, and we also found dose-dependent separation. But what we're seeing here is larger reductions in anandamide, plasma anandamide levels from baseline to day 4 in people who receive CBD, with the most pronounced reductions being in the 900 milligram group. So not only did this not support our hypothesis, this was opposite in a very sort of compelling way to our hypothesis. So what I wanted to do is I'll pause for a moment, right? Because I know we've got some people who spend some time in this space. I want you to think about like what might be going on here. I'll share what our post hoc hypothesis was as to what's going on here. But I'd love to hear people's input on this as well. So post hoc hypothesis, what the heck's going on here? So first I'll have to thank Jamie Caroon, who did a deep dive into the literature. Led me to a few studies that sort of triggered some thoughts about what might be happening here. The first is this idea that there's a study showing that acute stress in humans, healthy humans, increases plasma anandamide. And so this was a study that I'm showing you here that gave people the threat of giving a speech and showed that under the threat condition, anandamide goes up, meaning that when people weren't threatened, anandamide levels are lower. And so the question is during the resting phase, when plasma was collected, is it truly at rest? Remember, right, our population, people with social anxiety disorder, coming into the laboratory environment, meeting people they've never met before, a lot of unfamiliarity, is a stress-provoking environment. And there was this other study that did 4 consecutive visits where they had tested different doses of CBD and THC in healthy humans. And what they found is a progressive reduction in pre-treatment anandamide measured at every session that they thought may be related to participants becoming less anxious about the testing procedure. And so what we thought is that in this population with social anxiety disorder, and given what we saw in the speech task with the anxiolytic effects of CBD, maybe people on the higher CBD doses were less anxious just when they came into the lab. And that's what we found. So this is looking at change in anxiety at rest. And what we see here is, again, a dose-dependent greater reduction in baseline resting anxiety in people in the higher doses of CBD. So again, it's not, blowing us out of the water, but it's suggestive that maybe there was sort of this additional, I don't know if it's a methodological, sort of just a reality confound of a collection of plasma anandamide. So just to kind of wrap up and take this home, what about safety? Side effect profile comparable across the 3 arms. Severity of side effects was generally mild. Again, didn't differ between arms. And impairment, also generally there's no impairment, very little impairment due to side effects. If anything, slightly trending toward greater impairment in the placebo group, probably because these folks didn't have the benefit of the anxiolytic properties of CBD, so they were more stressed out about any side effects they perceived. All right, so to summarize, findings from this trial suggested that CBD dose-dependently reduces threat reactivity. It also suggests when you look at the dose range used in prior studies that maybe, for instance, that chronic dosing trial of 300 milligrams, that maybe have been underdosing people. And it suggests that the higher dose may be more efficacious. And I would like to actually thank the CMCR for not accepting our pilot proposals, because when I look back at that, we really hadn't designed the dose range to be optimal. And I think this data suggests that we can and should push the dose higher in patients with social anxiety disorder. We also found that CBD dose-dependently reduced plasma anandamide. Again, some questions as to why this might be. But when we think about that story linking anandamide to anxiety reduction, I think that story is becoming a little bit more murkier. And the reason is since we proposed the trial, there's been several completed phase 2 trials of FAAH inhibitors. And what I'm showing you here, this was recently published in Neuropsychopharmacology. This was a trial in post-traumatic stress disorder of a FAAH inhibitor relative to placebo. Again, reliably showed that anandamide increased in people assigned to the FAAH inhibitor. However, they found no differences in anxiety or really any of the other clinical symptom measures. And what that suggests to us is that you can modulate anandamide, but maybe it doesn't have that tight coupling with anxiety that we'd initially thought. So I think the story there remains a little bit unclear. Again, both doses of CBD were well tolerated, super high adherence in that short time frame. And I'd suggest that this is supportive of chronic dosing trials to take the higher dose forward and to evaluate its efficacy. Of course, you're wondering like, hey, how's the R33 going? What's going on with that? I can tell you that unfortunately, NCCIH did not continue this award because of the anandamide finding, but they encouraged us to go back in and submit a new submission. So that's where we're at. And with that, I'll end again thanking my team, the various support we received. And of course, if anyone wants to reach out after this, please feel free to do so. So with that, I think we've got a little bit of time for questions. Yeah, not too bad.